So before we move on to begin talking about nucleic acids, that was the other macromolecules that I want to draw our attention to. I want to finish up talking about proteins by describing their function as enzymes. Now what I want to point out is that not all enzymes are proteins. Most enzymes are proteins, but what we've learned recently is that there are many RNA molecules which can also function as enzymes. That is, RNAs can fold up in very specific structures analogous to proteins. They carry out reactions, and when we talk about the nucleic acids, I'll bring that up. But here we're going to be talking about proteins as enzymes, and what enzymes do is they serve to speed up reactions in cells. I'll give you an example in a minute of some types of enzymes. And there's three basic steps, and two of them I want to talk in a little bit more detail about. One is, and they basically work like this, they bind the substrate. Let's say the substrate is glucose, and we want to break down glucose. So it binds the substrate, and the binding is extremely specific, and then it facilitates the reaction that needs to take place, and then it releases the products. The substrates bind with extreme specificity. The enzymes are highly targeted to specific substrates. We'll look at that in a minute, and that depends on the positions of particular amino acid R groups. But let me just give you an example of some enzymes that people have studied for a long time. So this table just shows some common classes of enzymes. Most enzymes end with an ase. So a nuclease is something that breaks down nucleic acids. Protease is an enzyme that breaks down proteins. A polymerase is a protein that makes polymers, in this case of nucleotides into the nucleic acid. And then we talked about kinase. Kinase is what adds phosphates. Phosphatase removes phosphates from substrates like proteins. So this is a series of some common enzymes, but all of them speed up reactions that could, in principle, take place on their own, but they're not very likely. But they speed them up quite significantly. Now, the first thing I want to talk about is the binding site. So what has to happen is there has to be, on this particular protein, there has to be a specific site to bind the substrate, and that's called the substrate binding site. Sometimes it's referred to as the active site, if that's where the reaction actually takes place. And so a drawing might look like this. The green is the protein or the enzyme, and then you have the ligand or the substrate binds very specifically into the binding site of the protein. And what I want to bring out here is that, again, it's similar to what we've been talking about with respect to proteins, is the position of the R groups on the surface create a large number of bonds that provide the specificity. So again, it's the same kind of thing. It's the hydrogen bonding, or it would be the ionic bonding. It's not going to be covalent bonding, because covalent bonding would not release it, because those are bonds that are very difficult to break. So let me show you an example of a binding site that's been worked out for an enzyme that hydrolyzes what's called cyclic AMP. The reaction is not that important. What I want you to look at is the details of the binding site. So this shows the specificity of the binding site of cyclic AMP into an enzyme which ultimately will break some of the bonds in the cyclic AMP. But the point is the interactions with specific amino acids. So the cyclic AMP, it's illustrated in the center. It's kind of like this orange compound. And then within the green are showing specific R groups: serine, threonine, glutamic acid, arginine, and serine. And if you look carefully, what you see is the serine on the right-hand side, hydrogen bonding with certain parts of the cyclic AMP. Same as with threonine. Same as with the glutamic acid. And we're actually showing the hydrogen bonding with that O coming off the glutamic acid with the hydrogen. And you actually have right next to it, I didn't see that before, in gray is actually the participation of a part of a peptide bond. And then arginine is interacting with an ionic bond, and then the serine with the hydrogen bond because it has the OH. So these are amino acids that are positioned on the surface of the cell, the surface of the protein. And what I want to point out, again, is I'm driving this principle home, is that it is the precise position of these amino acids in the protein that gives rise to this specific interactions with this compound. Now, that, of course, is reflected in the primary sequence because these things have to be in a very specific position. But above and beyond that, the protein is folded in a very specific way, and that's due to the interactions of all the other amino acids in that particular domain. So you're seeing here just a few of the amino acids, but all the other ones are participating in causing this thing to fold up in order to create and bring those specific amino acid R groups into such a very precise position such that it would interact with this compound. And that's the same with all amino acids, or all enzymes, excuse me. Now what these enzymes do is they increase the rate of reaction. Let me just show you an example, a pretty dramatic example here. So enzymes increase the rate of reaction, or they increase the probability of a reaction taking place. They cannot carry out reactions that wouldn't normally take place naturally. They can't go against the rules of thermodynamics. What they do is they facilitate the reaction which would otherwise take place anyhow. So here's an example of the hydrolysis of urea. Urea is NH2, the C double bond O, NH2. In the addition of water, you get the hydrolysis to 2 molecules of ammonia and CO2. So this hydrolysis of urea will take place spontaneously. And we know the rate. The rate is 3 times 10 to the minus 10th per second, which turns out to be 1 every 106 years. One hydrolysis reaction every 106 years. So it couldn't happen, but it's extremely rare, extremely unlikely, but it will happen. But you add urease, which is an enzyme that degrades or hydrolyzes urea. All you do is you add this protein to the solution, and the new rate goes from 3 times 10 to the minus 10th to 3 times 10 to the 4th. That's 30,000 per second. So 30,000 urea molecules hydrolyzed per second. It's 100,000 billion increase in the rate of the reaction. So it's pretty clear these things increase rates of reaction of reactions that would occur otherwise, but extremely rarely or extremely slowly. So what's the mechanism of action? And that is, how do these enzymes speed up these reactions? What occurs in the active site that leads to such an increased rate? So there's four different aspects to this. All these participate in one way or another, some depending on the reaction that's occurring, maybe more prevalent than others, but here are some of the mechanisms that are involved. The first is increase the local concentration of the substrates. That is, if two compounds need to react, let's say two amino acids need to react to make a peptide bond. Well, if they're centimeters apart or millimeters apart, or in two different parts of two different locations in the water, it's just not going to happen because they're not together. So when you bring these into an active site, you're effectively increasing the concentration of the substrates. They're together now. The second is it orients the substrates in optimal positions to react. So in other words, in order for these reactions to take place, the substrates need to be positioned so that the points at which need to make a new bond, or let's say break a bond, need to be oriented such that this will occur. So they orient the substrates so they react more easily. Sometimes they participate in the reaction themselves. And that is, if a substrate needs to have a hydrogen added to it, that could happen coming from, let's say, a basic amino acid, or hydrogen removed as part of a reaction. That could happen. It can be added to an acidic group. So sometimes they can actually participate in the reaction themselves. But it seems like the most important aspect is the enzyme binds to what are called transition states. Transition states are states of the reaction, like part of the process of the reaction, that actually is extremely unstable and extremely difficult to create. They're unstable intermediates. That's the transition state. An unstable intermediate that is energetically difficult to create. And what happens is, in most cases, or in many cases, the enzymes have their R groups positioned so that they allow the transition state to occur more easily. So I'll give you an example of the hydrolysis of a peptide bond, part of a protein. So here we're going to look at the hydrolysis of a peptide bond, and of course peptide bonds are the bonds that link up amino acids in a protein. And so on the kind of the top left, you can see an R group. Then there's the double bonded ONH, that's the peptide bond, and then there's the next R group. Now this hydrolysis can, in principle, take place spontaneously. With the addition of water, which is shown in orange, the oxygen of the water will form a new bond with the carbon, with the release of the proton. And so that's what's shown on the right-hand side. The oxygen has made a new covalent bond with the carbon, and then you have an oxygen bound by a single bond, and then you have the NH as part of the peptide bond. This is the transition state. This is a state that is very unlikely to occur without some input of energy. It's a very unstable, difficult to create, and difficult to maintain. But once that transition state occurs, then with the addition of that proton, then you get a cleavage of the bond, where you see the products on the bottom. You have the C double bond OOH, which is the carboxy terminus of the amino acid, and then you have the NH2, which is the amino terminus of the other product. So the point is, the transition state, very difficult to create. It's a very odd kind of reaction. What happens, though, is the protease, in the reactive site, the protease stabilizes the transition state. So there are amino acids that are positioned so that will hold that OH and hold that oxygen on the top and allow it to occur more readily. And what the way it's phrased is it reduces the activation energy. So enzymes facilitate the creation of transition states and allow these reactions to take place more smoothly and more easily. Now I want to spend a few minutes looking at some relatively simple enzyme kinetics, which are classic processes in the way enzymes carry out their reactions. So this is a kind of classic prototypic graph of standard enzyme kinetics that is looking at reaction rates. Along the x-axis, we have the substrate concentration that is very low on the left-hand side and very high on the right-hand side. So you'd have a concentration, let's say it would go from 0.0001 molar to maybe 1 molar on the high side. And then on the y-axis, we have the reaction rate, and that is measuring how quickly the product of the reaction appears. So when you look at this, what happens is that the reaction rate seems to be dependent on the substrate concentration. So if you look at low to high, what you see is as you begin to increase the substrate concentration, the reaction begins to occur. And that makes sense in a way. If you don't have a substrate, you're not going to get reaction. But the more substrate you add, it's almost at the beginning, you almost get like a linear increase, so it's proportional. But then what happens is at one point, as you increase the substrate concentration, you can see the increase in reaction rate begins to slow down, and eventually it plateaus out, and it reaches what's called the Vmax. That's the maximal rate of the reaction. And the plateauing represents the reaching of saturation of the enzyme. What does that mean? The saturation of the enzyme, that means that all the enzymes in the solution are occupied by substrate. And they're working continually. So you can't make it happen any faster by adding any more substrate because all of the enzymes in the solution are functioning and carrying out the reaction. So you've reached saturation. At the beginning, it's kind of linear because you have excess enzyme in the solution. As you increase the substrate, you increase the reaction, and then it plateaus out. Now, there's a couple terms that are important, 3 terms. The Vmax represents the maximal rate of the reaction. And then we have a value called the Km. The Km represents the substrate concentration that allows the enzyme to reach 1/2 Vmax, half the maximal rate. The Km is the substrate concentration at which you get 1/2 Vmax. So in essence, what the Km represents is effectively the efficiency of the enzyme interaction with the substrate. How efficient does the enzyme interact with the substrate? So if you have, I'll give you an example of some drawings that I made in a minute, but if you have a high Km, let's say it's moved over to the right, and you look at the graph, that means that you would need a higher substrate concentration in order to reach 1/2 Vmax. You need more, so it's reacting less efficiently. If you have a low Km, let's say it's to the left, then it's saying it's reaching the 1/2 Vmax at a lower substrate concentration, and so it's more efficient in that sense. So let me show you a few examples. So this may help explain what this is. So this is one that I drew. So again, on the x-axis, you have the substrate concentration from low to high. On the y-axis, you have the enzyme rate. So let's start out by looking at enzyme A. So enzyme A, similar to what we saw before, you increase the substrate, you increase the rate, and then eventually you plateau out and you get the Vmax. So there you have a Vmax, and the 1/2 Vmax, you can see I have that dot on the line. That's approximately 1/2 the Vmax, and it has a particular Km. There's the arrow that goes down. Now let's look at enzyme B. Enzyme B, you have a lower Vmax, so the rate of the reaction is not as quick. But if you look at the Km, which is determined by the 1/2 Vmax, the Km is low. It's a lower substrate concentration, allows this thing to reach the Vmax much more quickly. And of course, if you look at C, you have the enzyme has a high Vmax, so it has a much greater rate of reaction ultimately. But yet, if you look at the Km, it takes more substrate to get it there. And so it has a high Km. So in essence, the more efficient enzyme is enzyme B, because it reaches the Vmax, or the 1/2 Vmax, at a much lower substrate concentration. You need less to get there. Let me just give you an example and if you have questions, you can contact me, obviously, or I think there may be some information in the book on that. But let's just I just want to give you an example of what kind of rates we're talking about. So here we're looking at the Vmax is the maximal rate of different enzymes. So urease, we talked about that earlier. That's actually one of the fastest enzymes that are known, hydrolyzed urea, 30,000 per second. Penicillinase basically breaks down penicillin, 2,000 per second. Beta-galactosidase cuts sugars, 200 per second. And the one at the bottom, this is the enzyme that fixes CO2, which I said was a very difficult reaction. It takes 3 molecules of ATP for each reaction. But this is the Rubisco, the ribulose bisphosphate carboxylase. Even with the enzyme, it only can do 3 per second. So it's a apparently very difficult reaction to take place. But this, of course, what fixes CO2 to give us all the compounds that we have on Earth, basically. So one of the ways to increase the reaction rates are by increasing the substrate. But there are 2 other ways I want to just show you. One is temperature and one is pH. So let's first look at temperature. So with temperature, you get a kind of an average effective temperature on an enzyme. So if we look at enzyme A, what you can see is, so the temperature is on the x-axis from 0 to 100. That's centigrade. 100 would be boiling, and then rate of reaction, that is how fast the products are produced. And if you look at enzyme A, what you can see is at 0 degrees, there's no reaction. As you increase, you get maximal reaction at around 37 degrees, which is body temperature. And then as you increase the temperature, the reaction plummets. So at 45 degrees, maybe 50 degrees, there's no activity at all. This is kind of like an average human enzyme. Most of the enzymes that are in our body work this way with respect to temperature because our body temperature is 37. So most of the enzymes will work at their optimal rate. What's happening here is, why the increase, why the decrease? So even though it's an enzyme reaction, it's still a chemical reaction. You still need energy. You need energy to allow this thing to happen. You need energy to provide the transition state, even though it's lower than it would be without the enzyme. You still need some energy, and the energy is provided by heat. And so as you increase the energy, you increase the rate of the reaction. What happens if once you go past that maximal reaction rate, is that as you're increasing the temperature, the enzyme denatures. You're breaking the bonds, you're breaking the hydrogen bonds, you're breaking the ionic bonds, and if the temperature is high enough, you can even break some of the covalent bonds. So that's why you get this decrease. Now, if you look at enzyme B, this has a similar profile, but the maximal reaction rate is much higher. And this comes from an organism that is basically stable under high temperatures. So you have these thermal stable organisms that exist like in the the geysers at Yellowstone National Park, or other deep sea organisms where the temperatures are very, very hot coming out of volcanic reactions. You'll have organisms growing there. So this is one that's called Thermophilus aquatica. And since the organism lives at high temperatures, then its enzymes have to be adapted to function at high temperatures, and so you see that. So this one here has a max temperature of around 80 degrees. And we know of some enzymes that have maximal reaction rate can be as close as 100 degrees, depending on where the organism originates. Now, the other variable that can affect enzyme reaction rate is pH. So here you have a graph which illustrates the effect of pH on enzyme activity. It looks similar in a way to what you see with temperature. You have an increase in the reaction rate, and then it's optimal at some specific pH, and then after that you get this decrease. And also you can have enzymes which function optimally at different pHs depending on what the origin of the enzyme is. So pepsin, for example, which has a very low pH optimum, that means it's acidic, is found in the stomach, where the conditions of the stomach are very acidic, and so any enzymes that are functional in the stomach need to be able to function at a lower pH. And here you have pepsin, which is an enzyme which degrades proteins, and the pH optimal is around 1 or 2, which is quite acidic. Then you have trypsin, which also digests proteins, but it occurs in the small intestine, I believe, where the pH of the interior of the small intestine is around 7 or 8. And so this enzyme is designed to work under those conditions, and so it has the pH optimal at around 7 or 8. Now you have an increase. If you take trypsin, for example, if you go from 3, which is acidic, up to pH 8, you get this increase in activity, and then it starts to decrease as you increase the pH above 9, 8 or 9. And the reason you get the increase and the decrease is really that the proteins denature under lower pH and denature under higher pH. It's not the same as with the temperature. With the temperature, you are increasing the temperature, which at the beginning causes an increase of energy in the reaction, and then you get the denaturation at the higher temperatures. Here, you get denaturation in both ways. So if you have an enzyme at pH 8, it works fine. As you make it more acidic, you denature the protein because it is changing the activity or changing the structure and function of some of the R groups that are basic and acidic. And then the same is true as you increase the pH, you change the activity of some of those R groups, which may be responsible for holding the protein together, so it denatures. Okay, so those are 3 things that affect enzyme activity: the substrate concentration, temperature, and pH. The last thing I want to mention is that many of the drugs that we use actually function by binding to enzymes and inhibiting them. And I'll just close with giving you one example. So I just want to finish up talking about enzymes by just mentioning that many drugs that we use are drugs that inhibit specific enzymes. These enzymes would be, a high expression of these enzymes may be responsible for particular illnesses or syndromes. And so just to give an example, so that the diagrams are not specific to any particular example, but just to give an example is an enzyme called HMG coenzyme A reductase. This HMG coenzyme reductase is, if it's overexpressed, is responsible for increased lipid fat levels in the blood. And so what often, if you have those increased lipid levels in the blood, that can lead to cardiovascular disease or heart attack, stroke. And so the idea is to try and lower the lipid levels in the blood. And that can be done a number of ways. It could be done by changing diet, which could be very important. It can be done by exercise. But in the cases where diet or exercise may not control, or if you have some genetic predisposition, then you might end up using what's called a statin, which inhibits the coenzyme A reductase, and two that are common on the market are Lipitor or Prevestatin. These are two inhibitors of the HMG coenzyme reductase. Now, there are two types of inhibition. There's what's called competitive inhibitor or non-competitive inhibitor. Let's start with the non-competitive inhibitor on the right-hand side. A non-competitive inhibitor, that's shown in the dark green in this drawing, will bind to the active site of the enzyme and bind so tightly that it can't be removed. Maybe there's a covalent interaction with one of the R groups on the amino acids, but it basically permanently blocks the enzyme. It cannot work anymore, no matter how much substrate you have in there. The competitive inhibitor is different, and these coenzyme A reductase inhibitors, the Lipitor or the Prevestatin, are competitive inhibitors because you don't want to totally block the enzyme. You need to have some lipids produced, but what it does is it reduces the activity, in a sense, of the enzyme. It reduces its availability, but it doesn't completely eliminate it. With the competitive inhibitor, you have, and that's a little bit lighter green, the competitive inhibitor can bind to the substrate, the enzyme binding site, the substrate binding site of the enzyme, but it's not permanent like in the non-competitive. It's a competitive inhibitor, and so what that means is that if you have enough substrate, it can replace the inhibitor. So there's an interplay that goes on. There's an equilibrium. So you can have the inhibitor, and you can bind to some of it. It can come off. It can carry out some of the reaction. So it doesn't completely block it, and it can be competed off. Okay, so that's pretty much what I wanted to say about enzymes. We've finished proteins. The proteins, of course, we spent a lot of time on because they are very sophisticated, very complicated, very interesting macromolecules. A lot to say about them. And we're going to move on next time and start talking about nucleic acids. All right. Have a good rest of your day.